Good morning, students. Today, let us learn about Mendel's laws or Mendelism. The laws of Mendel that explains the mechanism of heredity is called Mendelism. That is, the laws of Mendel that explains the mechanism of heredity is called Mendelism. Based on Mendel's experimental results, certain principles are framed. These principles are called Mendel's laws. They are as follows. Number 1, law of dominance. Number 2, law of segregation or first law of Mendel. Number 3, law of independent assortment. First, law of dominance. Law of dominance is formulated based on the monohybrid experiment. Each organism is formed of a bundle of characters, and each character is controlled by a pair of factors or genes like capital T or small t. Each of the paired factors is responsible for the expression of a particular character. That is, capital T is responsible for the expression of a tallness of the plant, and small t is responsible for the dwarfness of the plant. Mendel's law of dominance states that one factor or one gene in a pair may mask or prevent the expression of the other gene. I repeat the same. One factor or gene in a pair may mask or prevent the expression of the other gene. The character or factor that appeared in the F1 generation is called dominant character or dominant factor, and those which did not appear in the F1 generation is called recessive factor or recessive character. A recessive factor freely expresses itself in the absence of its dominant allele, and that is the essence of law of dominance. Second law, law of segregation or first law of Mendel or law of purity of gametes. Law of segregation is formulated based on monohybrid experiment. This law states that whenever a pair of factors for a character is brought together in a hybrid, they segregate or separate during the formation of gametes. I repeat the same. Whenever a pair of factors or genes for a particular character is brought together in a hybrid, they segregate or separate during the formation of gametes. That is, during the formation of gametes, the paired genes segregate or separate and enter different gametes. Hence, each gamete contains only one of the paired genes which are responsible for a particular character. Hence, this law is also called purity of gametes. Next, explanation for Mendel's first law. Mendel used a capital letter to denote a dominant factor or dominant gene and the corresponding small letter for the recessive factor or recessive gene. Mendel crossed a pure breeding tall pea plant with a pure breeding dwarf pea plant. According to Mendel, the pure breeding tall plants of the parental generations contained two identical genes for tallness. They may be shown as capital T, capital T. Similarly, the pure breeding dwarf plants contained two factors or genes that may be shown as small t, small t. During the formation of gametes, the factors or genes separate. All gametes of the tall plant will carry the factor capital T, and all gametes of the dwarf plant will carry the factor or gene small t. The zygote produced by this cross will have the factors or genes capital T and small t. Hence, all the offspring will have capital T and small t. All of them will be tall because only the factor capital T expresses itself. Small t does not produce any effect as capital T is dominant over small t. The tall plant with a gene content of capital T, small t of F1 generation during selfing will produce two types of gametes as the factors or genes capital T and small t will segregate or separate. This means that half of the total number of gametes formed will have the capital T factor or gene and half will have the factor or gene small t. When gametes carrying these factors fuse to form zygotes, there will be three combinations of genes, that is capital T or capital T small t or small t. Of these three, both capital T and capital T small t zygotes will produce tall plants. The small t zygotes will produce dwarf plants. Thus, a total of 3 by 4 will be tall and 1 by 4 will be dwarf. This is the typical 3:1 ratio observed by Mendel. Next, monohybrid test cross. It is difficult to identify the homozygous or heterozygous condition just by looking at the phenotype of the plant. In order to find out homozygosity or heterozygosity of a given plant, test cross is conducted. Here, the given plant with unknown genotype is crossed with a pure recessive plant whose genotype is definitely known. I repeat the same. Here, the given plant with unknown genotype is crossed with a pure recessive plant whose genotype is definitely known. For example, tall plant which may have alleles, that is capital T or capital T small t, which is not known, is crossed with a pure recessive plant with a gene content of small t. I repeat the same. A tall plant which may have alleles capital T or capital T small t, which is not known, is crossed with a pure recessive plant with a genotype of small t. In this cross, tall plant produces one or two types of gametes and a pure recessive plant produces only one type of gamete. A test cross produces two types of offsprings. 50% of them would show the dominant character and the other 50% would show the recessive character. The appearance of recessive character immediately shows that the given plant is heterozygous. I repeat the same. The appearance of recessive character immediately shows that the given plant is heterozygous. On the other hand, if all the offsprings are tall, that is 100% tall, the given plant is homozygous. The test cross ratio is always 1:1. That's all.